So in this video, I'm going to be going over some of the answers to the Chemistry of Life test review. 1. Living things are full of chemical reactions. For most of these reactions to take place, the reactants need to be dissolved in water. 2. Because water molecules are highly polar, they can form hydrogen bonds with other water molecules. This gives water its stability as a liquid. That's that whole idea. Because they stick together, they don't tend to fly off and evaporate, so it tends to stay as a liquid. When water molecules stick to each other, it is called cohesion. When they stick to other things, it is called adhesion. The ability of water to absorb a lot of heat without changing temperature much is called having a high heat capacity or a high specific heat. This is important for living things for two main reasons. 1, living things are full of water, so that allows them not to change temperature very easily. You want to stay at a stable temperature. And 2, the planet is mostly covered in water, and that allows our planet to stay at a more stable temperature and not fluctuate in temperature easily. If I'm going to draw a water molecule looks kind of like a Mickey Mouse shape, where the small 2 hydrogens and the larger 1 oxygen. The positive side is the hydrogen side and the negative side is the oxygen side. And then if I were to draw a second water molecule interacting, I want to make sure that the hydrogen side of 1 is forming a little hydrogen bond with the oxygen side of another one, because positive attracts negative, and so the positive part of 1 will sort of stick to the negative part of another one. Okay. Looking at this temperature map, things you should notice is that the coastal cities here, San Francisco and Los Angeles, have much lower temperatures than the more inland cities here, which are kind of in the hundreds. And the reason for that is because the coastal cities are near water, and water resists changing temperature. So it's not going to get as extreme temperatures in areas that are near water, like San Francisco, as it is in areas that are far away from water, like Las Vegas. Why is it a bad idea to throw water on a grease fire? So that's that whole idea where lipids like grease, fats and stuff, and water do not mix. So if you put water, which is polar, on grease, which is non-polar, the water will be repelled by the grease, so that water will not be able to put out that grease fire. In fact, it could make it worse. Ice floats in liquid water. Why? Ice is less dense than water. And again, this is unusual. If you recall back to the labtivity, the solid fat sunk in the liquid fat, but the solid water floats on the liquid water because ice is less dense than water. Right, standard 2 is about the organic molecules. A single building block of a large organic molecule is called a monomer. When they are put together, that is called a polymer. So 1 block, monomer, a bunch of them chained together, polymer. And most of our organic molecules are polymers. They're large molecules built out of many small building blocks that are chained together. CHNOPs are the most common elements in organic molecules, and those stand for carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Probably the most important element for organic molecules is carbon. That's sort of the framework that all our organic molecules are based on, because it can form 4 bonds. It can bond with 4 other things, and it is also very light and stable. Types of carbohydrates. Sugars. Sugars are used by both plants and animals. They're found in the bloodstream of animals. They're found in things like fruits and plants. Function of sugar is quick, immediate energy release. Starch, found in plants. Function is energy storage for plants. Cellulose, also found in plants. Its function is structural support for plants. So that is fiber, that is wood, that is what holds a plant upright since plants don't have bones. Didn't leave a lot of room for this, but hints that tell you an ingredient is sugar. Things that end in ose, you should always suspect of being sugar. Things that are labeled as syrups are sugars. Anything that is like a fruit puree or a dehydrated juice of some kind, that's going to be a sugar. If it's just straight up labeled sugar, that again is going to be a sugar. Honey, malt extract, those are also sugars. So there's a lot of ways you might see sugars on a food label. How are lipids used by living organisms? Many ways. You have fats that are used for long-term energy storage, cushioning your organs, and insulation, so keeping you warm. You have waxes like earwax that keep stuff out of your ears. You have phospholipids that make up your cell membrane, so those are the barrier around all of your cells. You have oils, which keep your skin moist and your hair. They also are used for animals that live in the water, like ducks and otters, for keeping water off of their skin for waterproofing. And then you also have the steroid hormones like testosterone, which are chemical messengers and are also made from lipids. Saturated versus unsaturated fats. Saturated fats have every carbon bonded to the maximum number of hydrogens. They are usually solid at room temperature, so this means no double bonds. They're kind of shaped like a zigzag. Unsaturated fats have some double bonds, and not every carbon is bound to the maximum number of hydrogens. They are usually liquids at room temperature, like oils. One characteristic all lipids have in common: does not mix with water. Another way of saying that is that lipids are all non-polar, whereas water is polar. The monomer of a protein is called an amino acid. Proteins are used in the body for lots of different things. I would look up the list of things you have there. Remember, proteins are not just for muscles. They do all kinds of different important jobs in the body. The elements contained in our three main organic molecule categories here. Carbohydrates have carbon, hydrogen, oxygen. Lipids also have carbon, hydrogen, oxygen, although they have sort of more carbon, hydrogen, and less oxygen. And then proteins have carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. And then the P, of course, comes in with the nucleic acids, which we're not going to have to worry about for this test. All right, standard 3, enzymes. Each enzyme only works with one specific substrate. The substrate binds to the active site of the enzyme. The enzyme lowers the activation energy, then the enzyme releases the products. And so if we label that on the drawing, in this drawing, this is the enzyme, the blue part. The orange is representing the substrate. This part right here where the substrate is binding to is going to be the active site, and then the end result of our reaction is going to be the products. So the substrate comes into the enzyme, binds to the active site, the reaction takes place. In this case, the reaction was splitting that substrate, breaking a bond, splitting it into two smaller pieces, and then the products are released and the enzyme is left the same as it was before, and it can carry out more chemical reactions, breaking up more of the substrate. Enzymes, remember, can only do one job. Each enzyme only performs one specific reaction with one specific substrate, and that is because, as you can kind of see in the drawing, the substrate and the enzyme must be a perfect fit, kind of like a lock and key. The active site and the shape of the substrate, and the charges as well, have to match up perfectly for the enzyme to be able to react with that substrate. So one enzyme cannot react with multiple different types of substrate. It can only react with one. And then here we have a graph of different pHs and how active these three different enzymes are at those different pHs. So if we look at this, we can see that pepsin, which is found in the stomach is active at a much lower pH. And that makes sense because the stomach is very acidic. And so that enzyme needs to be able to operate in a acidic condition. These other enzymes are found in the saliva, so in the mouth, and I believe in the intestines. And so those are less acidic environments, so they need to be able to operate, the saliva at a more neutral pH and the trypsin at a more basic pH. So basically, wherever the enzyme is meant to operate, it needs to be able to function at that pH and also that temperature of where it needs to function. So if we were to put this pepsin in an environment with a pH of 8, you can see that that is well outside of the range that pepsin works at. So what we would expect, the vocab word would be, this enzyme would denature. And what that means physically is, if the enzyme were shaped like this, if I put it in that pH that's outside the range it can operate at, suddenly this enzyme might be shaped like that. And that means our active site right here has changed shape. Therefore, it will no longer be able to fit its substrate, and so the enzyme will not work. A denatured enzyme can't function because the active site has changed shape, and so the substrate can no longer fit in the active site. pH is one factor that affects enzyme activity. What other factors? So a big one is temperature. Similar to pH, if an enzyme gets too hot, if it goes outside of its range of temperature it can operate at, it will also denature, just like this, and no longer be able to function. There's other factors. You could affect enzyme activity by adding more enzyme or adding more substrate. There are things called inhibitors, which can also slow down an enzyme. The main ones that you need to remember about are pH and temperature, though. All right, that concludes this key to the test review. If you have any further questions, please ask or send me a Schoology message.